Okay, thank you for the introduction. Yeah, I'm Nurdan. Currently, as you mentioned, I'm working as postdoctoral researcher in the CP lab at Cold Spring Harbor Laboratory. But in my presentation, I will explain you the phylogeny approach we developed during my previous postdoc in Adebari lab. Okay, so for my project and for the topic of this talk, the focus will be how to interpret the effect of a missense mutation. As we already know, single nucleotide variants might have no effect on protein function, but also they can alter the protein function and cause disease. Because of its clinical importance, of course, there are many tools presented in the literature. Sorry. For example, DBNSFB is a comprehensive database of including the predictions of many computational approaches. And in version 4, it reported it includes like 45 prediction algorithms and 9 conservation scores. So why we need another one? Because we believe that existing approaches mostly overlook the evolutionary relatedness and phylogenetic information. Most of the existing approaches are benefiting from like using multiple sequence alignment as the measure data. But we believe that by ignoring evolutionary relatedness and just using multiple sequence alignment alone, like causes the redundant counting of the dependent substitutions. And I will try to explain you our motivation in that slide. So MSA the master data for many tools, but is it enough? I'll try to convince you that it's not enough. Let's assume that we have 5 positions. Think that each of these columns are a position from your multiple sequence alignment. And our goal is to predict whether a substitution from D to Y at human is pathogenic or not. Here you are seeing human on the first row. For the first position, it is easy because the position is conserved during millions of years through evolution, which means most probably this position cannot tolerate any alterations. So we can comment that probably a substitution from D to Y will be pathogenic. But the situation is not that easy for many cases. Let's check second and third positions. If you are just seeing multiple sequence alignment, and if you are just using frequency or conservation, these two positions are exactly equal for you because you are just observing Y one time, and all other species are observed as D. But when we check the phylogenetic tree, we are seeing that for the position 3, the Y is observed at a really close species to human. The branch length is small, and which means probably these sequences are kind of really similar to each other. On the other hand, for the second position, for the second column, the Y is observed at birds, which is both with respect to tree far from human, also branch length is long, which means you can already expect a substitution mutation on that branch. So if you want to comment about these two positions, to discriminate these two positions, MSA is not enough. But by checking phylogenetic tree, we can say that most probably this Y will be tolerable in human too, because if gorilla can tolerate it, which are really their sequence is similar to human sequence, most probably human can tolerate as well. However, for the birds, we cannot guarantee anything and we cannot say anything related to neutrality. So the first important information we can gain from phylogenetic trees is sequence relevance, whether the mutations occur at relevant sequences or not. And now let's check the fourth and fifth columns. This time, again, if you are just seeing multiple sequence alignment, you can say that the frequency is Y is observed 8 times, all the remaining parts are D. So again, these two positions are exactly the same in terms of if you are just seeing multiple sequence alignment and just using frequency. However, for the fifth position, all of these Ys are coming from one single clade. We call them as phylogenetically dependent substitutions. Most probably there is a mutation here and D turned to Y. So it's not surprising to see that all children nodes and leaves are turned to Y. So all of these 8 Ys are resulted from one single alteration. And we don't want to redundantly count the effect of same mutation repeatedly. And for the fourth position, what is the difference? The number of Ys equal for position 4 and position 5, or column 4 and column 5. However, for the fourth position, Ys are both observed at closer species to human and also at different time points independently, which means during the evolution, many different species tolerated this kind of substitution, so which highlight the neutrality of Y in terms of human as well. However, for the fifth position, we cannot guarantee that because it is almost the same with the second column. There is just one change to Y, and it is just like change once. So we cannot guarantee anything related to neutrality. So second important information we gather from phylogenetic trees is dependence or independence of substitution. So if we can develop an algorithm, both considering the sequence relevance and as well as detecting the exact location of alterations and try to understand whether the substitutions are dependent or not, we can have an accurate algorithm. That was our hypothesis. And here I explain you the motivation of our approach. And now I will show you some real world examples. You are seeing a variation from like clean var. By the way, we also check the allele frequency with respect to nomad allele frequency. This variation is neutral as a change from R to H. But SIFT says that it should be pathogenic. SIFT is, as you know, a popular, like well-cited tool in variant effect prediction. Because SIFT says pathogenic because it's just observed twice. And like they are checking, SIFT checks the position diversity and the observed frequencies, and it thinks that it is low compared to like the reference amino acid. However, when we check the phylogenetic tree and alignment, we are seeing that both H's are observed at independent time points, and also one of the A's are occurred at a really close species to human. So as you see, the branch lengths are almost zero and they are kind of next to each other. So if lemur sequence can tolerate this, human might tolerate as well. I am not saying that we should assign the highest score, but we should include this kind of information, whether a relevant sequence to human tolerated this mutation or not. So if we can design an algorithm by using two important information I mentioned, we can easily classify this variant as neutral. And the second example is from clean var again. This time a substitution from S to I, isoleucine, is labeled as pathogenic with respect to clean var. And here you are seeing multiple sequence alignment and tree together. Both SIFT and polyphen2 says that this variant should be neutral. And when we check the alignment and tree, here, by the way, dark blue is S, the reference amino acid, and red is the alternating one, the I amino acid, isoleucine. The reason SIFT and polyphen2 saying neutral is there are many I's observed. But when we check the tree, we are seeing that most of these I's are dependent substitution. Most probably there is a substitution somewhere here. That is why we are seeing many I's. So it couldn't pass the first filter of neutrality. Secondly, we said that the sequence relevance is also important. As we see, it occurred far from human. Here the leaf of human, here the location of amino acid alteration. So if we can design an algorithm with our idea, considering our hypothesis, we can easily classify this variant as pathogenic because the location is far from human and most of them are dependent substitutions. So by motivating from this hypothesis, we developed PHECT, which stands for Phylogeny-Aware Computing of Tolerance for Missense Mutations. We published PHECT 3 years ago at MBE, and we made it to the cover on the August one. And now I can explain you details of our approach. PHECT requires three important inputs. The first one is multiple sequence alignment. I'm not saying that multiple sequence alignment is not important. Of course it is, but we believe that if you are just using multiple sequence alignment, by ignoring evolutionary information, it can be misleading. And the second important information, the second input of PHECT is phylogenetic trees, as I mentioned. And the last input is the ancestral reconstruction probabilities. As you already know, ancestral reconstruction helps us to go back in time and to predict the ancestral amino acid at these internal nodes. Because remember, the idea of our approach, we want to detect exact location of amino acid alteration, whether it is here, here, or here. And to do that, we need to know what's happening at these inner nodes. And we are doing it by using ancestral sequence reconstruction. Ancestral reconstruction gives us the probability distribution for each amino acid for each internal node. So for example, here, the probability of A is 1, the others are 0, or some distributions for each node, which help us to detect the exact location of amino acid alterations. So we have three major inputs, and now I can explain the working principle. Starting from human leaf, we travel through the tree by checking probability differences. The idea is try to detect where the probability of a specific amino acid increase. For example, if amino acid A is increasing from this node to that node, it means we can expect a substitution here. So our goal is just detecting the positive probability differences on the traveling process. We are repeating this for each amino acid and collect positive probability differences. And we construct a matrix like that. Here you are seeing like for amino acid A, we travel the tree. Here, these are the positive probability differences. Of course, the amount is also important. It's not the constant, just 1 or 0, but we take the amount of change, like 0.6, 0.3, etc. And we travel tree for R, and V2, like for each of the 20 amino acids, we travel through the tree and collect positive probability differences. And individual amino acid score is weighted summation of these positive probability differences. Why weighted? Because remember, a substitution at gorilla or bird is not the same for humans. So if the location of change is far from human, its contribution to the tolerance score should decrease. If it is closer, we should increase the effect like in terms of the tolerance score. So when the distance to human increase, we decrease the weight of this positive probability difference. So for each amino acid, the individual score is weighted summation of positive probability differences. And the final fact tolerance score is score A. This is coming from the tree traversal step. It is different for each amino acid plus diversity. Diversity means, for example, if you are observing, if your position is variable, if you are seeing 15 different amino acids in a position, most probably the remaining parts might not have any like effect in terms of pathogenicity either. So we try to combine the individual level score of amino acids plus position dynamics by using diversity over tree size, because definitely there will be a bias through the tree. Like if there are more number of nodes, collected amount of score might increase. That's why we balance the score, we normalize the score with respect to tree size. So that is a score, fact tolerance score for any amino acid A. And we compare the performance of fact with, of course, like the well-known and most cited tools in the literature, SIFT and polyphen-2 initially over 600 and more than 600 proteins. And as you see, in terms of AUC, FACT clearly outperforms SIFT. We run SIFT in two versions, like with our MSA and with its own MSA. And polyphen-2, like it outperformed clearly in terms of AUC, and I didn't put, but in terms of AUPR values. And here on the right-hand side, you are seeing the comparison of FACT with all existing conventional statistical approaches reported in the DBNSFP. Why I'm saying conventional? Because like currently FACT doesn't have any learning process. It's just a kind of probabilistic approach scoring the tolerance of an amino acid on a position. And we want to compare with the same level tools in the literature, and we pick those tools from DBNSFP. And as you see, in terms of both AUC and AUPR, FACT clearly outperforms all tools, except like it slightly outperformed ListTest2 in terms of that measures. But we also did different experiments over different test sets and also like different measures by like MCC, F1 score, and balance accuracy. In all comparisons, FACT clearly outperformed not only ListTest2, but also SIFT and polyphen-2 for these three different measures. So we show that FACT outperforms the other traditional statistical approach reported in DBNSFP. But as I mentioned, FACT doesn't have any learning process, and it might be limited in terms of understanding the nonlinear relationship between some different features, different components affecting variant effect prediction. So we developed FACTboost. And again, we published it at MBE last year, and we made it to the cover, luckily, on that year as well. And here, our novelties are input features. This time, FACTboost uses a gradient boosting-based learning model. And as I mentioned, the novelty coming from our input features. We took more than 400 features, including some features based on multiple sequence alignment, like frequency of observed amino acids, position diversity in terms of MSA, etc. Phylogenetic tree-related features like tree size, total tree length, and ancestor reconstruction-based features, And amino acid properties also another features, as well as fact score itself and component of fact. Like what I mean with component, because fact have like different process, like scores coming from treat reversals, score coming from the position diversity, etc. All of these components are taken as a different input feature in FactBoost. And we trained our algorithm, our model on like more than 13,000 human proteins with more than 7,000 variants coming from CleanVar, the UniProt's database, Homo sapiens variations database, and NOMAD. And here you are seeing the comparison of FactBoost with existing tools, all 40 tools reported in DBNSFP. I'm just showing some representative ones because like some tools have different versions. We just picked the best performing ones. And here I am showing the delta AUROC values, which means the tool AUC minus FactBoost AUC. So if the value is negative, it means FactBoost is outperforming the tools in comparison, which is the case in all comparisons. Here, I want to mention one more thing. As you see, we have different test sets, TS1, TS2 to TS5, and the difficulty level increases from TS1 to TS5. What I mean is, for TS1, like the classical approach did in the literature, what we do is we pick a date, like January 2020, and all variants added to CleanVar before this date is taken as training, and the new variants compared to this date is like kept as test set. So TS1 includes all the variations. TS2 is just includes unseen positions, which means if a position, not the variant, includes in the training set, we eliminate all variants at that position from test set are removed from our test set. For TS3, if we are just taking unseen proteins, what I mean is if a protein exists in the training set, we just eliminate all variants related to that protein from our test set. So as you see, the difficulty increases step by step. And what is TS4 and TS5? We call these as hard cases. What we do is like we just pick the variants, tools disagree. Because in terms of clinical perspective, these are the most important variants. Half of the tool says neutral, half of the tool says pathogenic. What should you do? Which one you should trust? So we specifically pick that kind of variants. And our threshold is 25%. If like more than 75% of 40 tools from the DBNSFP like agrees on a variant, we didn't keep them, we eliminated them. We just kept the ones that tools disagree. By the way, while constructing TS4 and TS5, we didn't consider the real label, or we didn't consider fact and FactBoost scores. But just what we do is we just take the predictions of 40 tools from DBNSFP and check whether they are disagreeing on a variant or not. And more importantly, we observe the highest gain coming from FactBoost over this harder case, which highlight the success of FactBoost in terms of variant effect prediction. And also, we want to compare with some novel and well-studied approach, well-performing approach in the literature, and specifically we compared FactBoost with alpha-missense, EVE, and CPT1. Again, TS1 to TS5, the difficulty level increased from TS5, and for in all comparisons, FactBoost performed better than the tool in comparison. And again, more importantly, especially for harder case, we are seeing a clear difference between FactBoost performance and the other tools' performance. I just put the AUROC values, but we are observing exactly the same trend for AUPR values as well. Okay. In the next step, we want to check like how each feature contributes to the prediction. And that's why we like computed the Shapley values. And the exciting thing for us was fact score is the most important feature for our FactBoost algorithm, which highlights the importance of our algorithm design. And also another important observation is 15 out of 20 most important features are tree-based, which means like because our hypothesis was we need to use phylogenetic trees and evolutionary information for more accurate prediction. And this result supports our hypothesis, and 15 out of 20 most important features, by the way, we cluster features based on the source of the like feature. If it is computed over MSA, we classify them as MSA features. So 15 out of 20 most important features are tree-based, which says that we need phylogenetic information and evolutionary relatedness to accurately classify missense mutations. Okay, until now, everything is working perfectly, but you might ask whether there isn't variance FACBoost misclassify. Of course there is, and we try to catch a pattern among these variants. Firstly, we check the mislabeled pathogenic variants by FACBoost, and we saw two trends, honestly. First one is here. Here you're seeing the first row is misclassified pathogenic variants by FACBoost, and this second set is the all pathogenic variants existing in TS1. And almost 96% of the pathogenic variants mislabeled by FACBoost is submitted to ClinVar without any guideline. As you already know, ACMG kind of provides some guidelines to label variants, like based on some population level evidence, clinical level evidence, even computational level evidence. Most of the variants in the ClinVar submitted without any guideline. I agree, but here the ratio is 9. However, for the misclassified pathogenic variants, the ratio increased to 25. So we wanted to check whether misclassification is associated with guideline status. And we checked with chi-square test, and the chi-square test came as like statistically significant, which says that if FACBoost cannot classify FACBoost misclassified the variants added to ClinVar without any guideline. So honestly, for this part, we don't blame FACBoost, but we start to suspect whether there is a problem with that variants submitted without any guideline. And as I mentioned, there are two trends. This second thing is something we can improve FACBoost, and we are working on it. I will explain you over an example. We realize that we are missing this kind of variants. It's not that frequent, but we want to understand what is the reason. For this uniprot ID, protein 65th position, a substitution from G to W is labeled as pathogenic with respect to ClinVar. But both FACT and FACBoost confidently says that it should be neutral. And we checked the alignment and tree. We observed that W's both observed at independent time points repeatedly and also occurred at closer species to human. So it should be neutral with respect to our hypothesis. We go back to ClinVar. We saw that it is a 2-star variant, which is like they are confident about it. That's why we started to search other positions in the same protein. And we found another position. Whenever this first position changed to W, a different thing from the reference, the second position also changed a different amino acid from the reference one for all three positions, which means these sequences can tolerate W because they have another mutation, most probably compensating the effect of this W, which is definition of coevolution, like organisms or like protein positions kind of evolve together to protect the protein function and integrity. So we suspect that there can be a coevolution signal here. And we check the 3-dimensional structure. Really, these two positions are most probably interacting because they are also close on the 3-dimensional structure. So FACBoost misclassified this variant because currently we are not considering coevolution. We just assume that each position is independent, like many existing tools in the literature. I know there are some tools considering coevolution, but the ratio is really low. There's like EV mutation, GEM, and like Rhapsody. But when we check in general, most of them are ignoring coevolution like us. But in our current work, it's kind of an ongoing work. I will not go into detail, but we divided this problem into two. Firstly, developing a coevolution algorithm by considering our idea, which is like using just phylogenetically independent substitutions and ignoring the just using multiple sequence alignment. And the second step was combining this coevolution algorithm with variant effect prediction. And we already solved the first part. 3 weeks ago, our algorithm FACE, again, the same like naming structure, which stands for phylogeny-aware detection of coevolution. 3 weeks ago, it's published at MBE again. I will not go into detail of the algorithms, but if you're interested, you can scan the QR code and feel free to contact me if you have any questions or comments. I will just mention that we compare phase with existing popular MSA-based approach like DCA, Gauss DCA, psycho, and mutual information. This phylogenetic tree-based approach like CAPS and COMAP in terms of different measures, not only AUC, but MCC, AUPR, or F1 score, we show that phase statistically outperforms other tools in coevolution prediction. So our next step is combining FACT and FACT-Boost with coevolution information and making coevolution-aware variant effect prediction. So in conclusion, we say that our key contribution somehow is we can design like more accurate algorithms by using phylogenetic information, and we offer a way for novel use of phylogenetic trees. And as I mentioned, our major ongoing works are combining FACT and phase, FACT-Boost and phase, and making coevolution-aware variant effect prediction. And also currently a master student in Adebali lab, they are continuing to work on this variant effect prediction problem. Currently, a master student is focusing on generalizing FACT for non-coding regions, and which they call FACT10. With that, I want to conclude my talk. I want to thank again to Variant Effect Seminar Series organizing committee for inviting me, and also I want to thank our funders as well as Adebali Lab members. And thank you for listening.
All right, thanks, Nurdan, for a really exciting and interesting talk. And thanks to the audience for putting some questions in the Q&A. So first question is whether this approach could be used not just for missense variants, but also for insertions and deletions.
Honestly, currently we didn't do anything related to insertions and deletions. Also in tree construction and ancestor reconstruction parts, we are using RAXMLNG and IQ3, and they are just ignoring the gaps. So currently we are not considering it. First, the coevolution algorithm considers that, but currently we didn't combine them. So I don't think we can just use for missense variants for now.
Great. And then the next question asks about distinguishing between effects for benign versus rare variants. So a lot of variant effect predictors are really bad at identifying benign variants when they're particularly rare in the population, and this could be an issue for phylogenetic approaches. So have you thought about or tried to evaluate the separation of pathogenic from benign when both are rare?
Yeah. I didn't get the question exactly. Could you repeat it? Sorry.
Sure. So I think it's asking about how, if you have benign variants in the population, that like a variant that's rare, evaluating whether it's actually benign versus pathogenic is really challenging because it's just not, you just don't see it very often in the population. So I guess maybe another way to ask the question, or maybe how I'm thinking about it, is have you looked at allele frequency or the allele frequency spectrum within your classification algorithm?
Oh, I got the question. Honestly, our trees are kind of like species level, not species trees, but gene trees. So we are checking whether observing the same amino acid at other species, other sequences or not. Currently, we are not doing a population level thing, but try to understand for general humans, this substitution is acceptable or not. In terms of allele frequency, we are not taking it in like as input feature or anything else, because currently while deciding the benign variants, we use allele frequency to label the variants as benign or not, and we are checking on them. So if the allele frequency is high, we label them as benign, and we try to predict this kind of variants currently.
Got it. Thanks. Maybe I'll end. So I'm not a coding variant person. I'm kind of a non-coding variant person. So my question was asking whether you guys are extending this approaches to non-coding variants, and that's the last thing that you talked about. So I was wondering if you're able, I know it's someone else's project, but if you're able to kind of share how you guys are thinking about that.
Honestly, currently we are following the same approach, but the non-code nucleotide level, she's currently applying the same approach exactly for different chromosomes. As far as I know, the gaps are causing a little problem there, so she's trying to handle that problem. But in the current version of approach, she's just applying a version of FACT, honestly, and it seems working pretty well.